Hello, and welcome to my first video on identifying and characterizing wild yeast strains. So far, I have isolated 3 wild strains from my backyard, and the first thing I'd like to do is figure out what species they are. So, in order to do that, I have to sequence a portion of their genomes, namely the ribosomal region, and compare that sequence to known sequences that are in the public databases. So, I'm going to show you how you can browse genome data and design primers for PCR and sequencing. So first we have to go to the NCBI website. The NCBI is the repository for genomic information. And click on the link Genomes and Maps. Under this link, click Assembly. And then here, enter the name of the organism whose genome we're interested in, which is Saccharomyces Cerevisiae. The first hit that comes back is Saccharomyces cerevisiae, strain S288C. So this is the laboratory strain of yeast, and it is the complete reference genome. Now what we're interested in is this refseq assembly accession number. I'm going to plug this number into my bioinformatics software, and that is going to tell the software where to go and download the genome data from. The software that we're going to use is UniPro Eugene. Eugene is a free and open source bioinformatics software that runs on Windows, Mac, and Linux PCs. So here I have Eugene opened. Now if you're running Eugene for the first time, the first thing you have to do is create a new project and give it a name and where you want to save it, and then click Create. I've already created a project called the S. cerevisiae Genome Project, and that is already open and loaded here. The next thing that you have to do is get the genome data. So go to File, search NCBI GenBank, and paste in the accession number that we got on the previous page, and click Search. Now, this search has given us all 16 chromosomes in the yeast genome from 1 through 16. Now, if you haven't downloaded these yet, you would click on Download, and it will save them to your save a copy of them to your local computer. I have already downloaded them, so I'm just going to hit Close. Clicking on the Project tab over here, you can see that I've got all of the 16 chromosomes in my project, so now I have to load them. And now that they're all loaded, let's take a look at what this genomic data looks like. So we have 3 windows here. The top window is a bunch of colored lines, the middle window is a bunch of letters, and the bottom window is a bunch of collapsible lists. The top window is a graphical representation of the genomic data, and the number line goes from 1 all the way up to 948,066. So this is too much data to take in one window, so let's zoom in on it a little bit. So I'll go highlight that region and then click Zoom to Range. And now we can get a better feel for what these windows are representing. So on the left, you'll see a bunch of metadata categories. We have centromere, we have gene, we have mRNA, ncRNA, which stands for non-coding RNA, tRNA, which is transfer RNAs, and so forth. The 3 most common types of categories that we're seeing here are mRNAs, genes, and CDS, which stands for coding sequence. So let's click on the gene for CDC60. When I clicked on the CDC60 gene, the middle window and the bottom window both shifted. So in the middle window, you can see this is the actual base pair level data. So this is the number line representing the position on the chromosome, and the top layer of letters, the top row of letters is the top strand of DNA, and this layer of letters is the bottom strand of the DNA. And when I clicked on CDC60, you can see that it started highlighting at this ATG, and the highlight continues on to the right. The bottom window contains all of the metadata in that tag. So it's highlighted the CDC60 metadata, and you can see that that's the locus tag, that's the gene for CDC60. I can click on the mRNA annotation, and it'll jump down to the mRNA annotation down here, and you can see all the information that's inside that one. And the most information is contained in the CDS metadata. And click on the CDS for CDC60, It gives you the gene name it's associated with. It gives you a note saying what the gene is. This one ligates leucine to the appropriate transfer RNA. And it also gives you the translated sequence, or the protein sequence of that gene. Now, this protein sequence here matches what we see highlighted in blue down here in the high-resolution window. So that's what these letters are that are above and below the DNA. These are protein translations, or these are the amino acids that the DNA here is encoding for. So the other thing to notice is that on the top row we have arrows pointing to the left and to the right. For CDC60, the arrow pointing to the right, the gene is encoded in the top strand of the DNA. But in this one, BEM4, the arrow points to the left and the gene is encoded on the bottom strand of DNA. So this is a brief overview in how to browse genomic data. To identify species, we're interested in looking at ribosomal genes. So we're going to have to filter through this metadata to find the ribosomal genes. Now, if I knew the gene name, I could type the gene name over here into the filter box. However, I don't remember the gene names, so I'm going to search for something else. Now, I know that the ribosomal genes all make ribosomal RNA, so that's what I'm going to filter for, rRNA. And all of the elements that contain rRNA are now filtered for. Now, this category, rRNA, are all located on one chromosome. I believe this to be chromosome 12. So if I click on that, we now get a new window open. And we can see that all the rRNAs are clustered right here in the middle. So let's go zoom in on that section. So here we can see that all the rRNAs are made up of these RDN genes, RDN25, 37, 18, and you can see that we have repeats of them. We have -1s and -2s, and that's because there's two copies of the rDNA genes, or the rRNA genes rather, in the yeast reference genome. So this is the first copy right here, and this is the second copy right here. So to simplify things, let's just focus in on the first copy. So I'm going to click on RDN37-1 and zoom into that range. This is a good time to explain why we're looking at the yeast reference genome when we're interested in identifying the species type of unknown wild yeast. This is due to the nature of the ribosomal genes. Some of them are very highly conserved throughout all yeast species, and others are highly divergent throughout all yeast species. Ribosomes are an essential piece of cellular machinery that's involved in protein synthesis. Some of these genes encode structural members of ribosomes, and others do not. For instance, the genes RDN18 and RDN25 encode for structural members of the ribosome. In fact, if we click on the annotation for RDN25 and take a look at its note, it says that it is part of the 25S ribosomal RNA, which is a component of the large ribosomal subunit. That means that anything that's part of the ribosomal subunits is going to be very highly conserved throughout all yeast species. But if we take a look at these ITS2 or ITS1 genes and their annotations, we can see that these are non-coding. They're not part of the ribosomal structure. What that means is that these ITS genes are going to be very divergent because they're non-coding, while the RDN18 and RDN25 genes are going to be highly conserved because they are components of the ribosomes. This is highly advantageous because it means that we can design PCR primers that bind to this end of RDN18 and this end of RDN25, and they'll work for all yeast species. And because these ITS regions are highly variable, this is going to provide sort of like a species fingerprint amongst the different yeast species. So what we need is PCR primers that are anchored in RDN18 and RDN25, and that will amplify both ITS1 and ITS2. Then we can sequence that PCR product and figure out what species of yeast we have. Now moving forward here, I would like to stop working on this entire chromosome 12 and just focus in on the region that I'm interested in designing primers for. So I'm going to select RDN37, and I'm going to go extract it into a new file. So export selected sequence region. I'm going to save this as a GenBank file, which is the same format as the files I downloaded from NCBI in the first place. I'm going to call it RDN37, and I'm going to save the complement strand. The reason I'm saving the complement strand is you can see all of these genes, RDN18, 25, ITS, and so on and so forth, are pointing to the left, which means that they're on the complement strand. So I'm going to export it, and now we have just that piece of exported DNA, exported data in our file, in our window here. So the first thing I want to do is clean out some of these annotations that I'm not interested in for designing primers, because it just kind of clutters things up. And actually, everything is cluttered except for the genes, so I'm going to remove everything except for the gene annotations. So again, we want to design primers that are anchored in RDN18 and RDN25. So ITS starts at 2501, and ITS2 ends at 3251. So the difference between 2501 and 3251 is 750 base pairs. So that's about 750 base pairs right there. So in order to design primers, I'm going to have the computer design the primers for me. So Eugene integrates the primer choosing algorithm, which is excellent. I've always had very good success using primers designed by Primer 3. So the first window we have to fill out is the targets window. The targets window says I want my targeted product to start at base 2500, and I want it to be 750 bases long. And the next thing I want to do is say, how big should the product be? So the minimum size of the product should be about 800 bases, and the maximum size, I'll say, is about 1200 bases. And pick primers. And right away, Primer 3 has chosen 5 sets of primers for me to use, to choose from. So we have pair 1, 2, 3, 4, and 5. So I'm sure that pair 1 is going to be fine, so I'm just going to remove these. And here we can see is pair 1 is the left primer, and here is the right primer. And you can see that they both span the ITS1 and ITS2, and they're both anchored in RDN18 and RDN25. Now, those of you watching this video who know about testing for species by sequencing the ribosomal DNA regions know that there are already primers that exist for this purpose. In fact, if we go to this website from Duke University, we can see that here's a schematic of our ITS1 region here and ITS2 region here, and here are the 2 primers that are commonly used, ITS1 and ITS4. These primers were reported way back in 1990, and we can actually map these primers onto our sequence. So I'm going to copy the ITS1 primer, and if I click on this little sequence search tab over here, I can enter that primer, and look, it's found it and it's highlighted the region right over there. And I can actually create an annotation for this. So I'm going to call this a primer. It is a primer, and I'm going to call this ITS1 and create annotation. So there we have our ITS1 primer. And let's do the same for ITS4. So there's ITS4. I'll change this name to 4 and create annotation. Now let's zoom in on the region where our primers are at. So I have some concern in using the traditional ITS1 and ITS4 primers. And that concern is due to the fact that they're really close to the beginning of ITS1 and the end of ITS2. I know from experience that sequencing data within the first 50 base pairs from the end of a primer tends to be a little bit unreliable. And then it gets more reliable from about 50 base pairs out to probably 750 base pairs. Now, if you take a look at the end of ITS1 and the beginning, or the end of the ITS1 primer and the beginning of the ITS1 gene, we're only about 12 bases away. So that means that the beginning of this ITS1 could have problems with the sequencing data. And if we go to ITS4 and the end of that one to the end of this one, we're only at 37 bases. So again, getting good sequencing data at the beginning of this region and the beginning of this region from these two primers might be problematic. Now these primers here, that primer 3 picked for me, gives us 96 base pairs of a buffer, and it gives us an even larger 226 base pair buffer from these two. So I know that I'm going to be getting high quality sequencing data when I enter the ITS regions. I also know that I'm going to get about 750 bases of reliable sequencing data from both of these primers, which means that this one will take me out to about here, and this one will take me out to about here. So I know that I'm going to get reliable sequencing data from both of these primers for the whole ITS1 and ITS2 sequences. And empirically, I've always had success using primers that primer 3 has chosen for me, so I'm inclined to use those. So the final step is actually ordering the primers. So this is quite simple. So let's start on the primer on the left. So let's just select it and then copy it. So now it's time to place the oligo order. I'm going to order my oligos from Eurofins Genomics. So to do that, we have to click on the Oligosynthesis tab. An oligo is short for oligonucleotide. An oligonucleotide is a single-stranded piece of synthesized DNA, and that is what is used for PCR and sequencing primers. So under this menu, we'll click Order Oligos. And we're going to do manual entry, and I'm going to order 2. So here I'm going to paste in the sequence for my ITS left primer, and I'm going to give it a name for ITS left. And down here we're not going to have any modifications. The only thing I'm going to change is the scale. I would like it to be 10 nanomolars, the smallest scale available. And now to get the next primer, we'll go back into Eugene, highlight our ITS right primer, and copy it, and paste its sequence here. And this is going to be ITS R. And I'm going to make this, again, 10 nanomoles, and add to cart. So now I'm going to check out, and I should receive the primers in the mail within a couple of days. Be sure to follow future videos when I'm going to run the PCR on my wild yeast strains and send the PCRs off to be sequenced, and then analyze the sequencing data in order to identify the species.